Please be seated.
Hereby, I open this academic ceremony in which Monize Caiado Decarli will defend the academic thesis Bioprinting of spheroid-based cartilage constructs from engineering to biological optimization aspects. May I invite you to present a summary of your study and the conclusions of your thesis. The floor is yours.
Thank you. Good afternoon, dear Prorector, members of the committee, supervisors, family, and friends. It's a pleasure to be here presenting my PhD thesis entitled Bioprinting of Spheroid-based Cartilage Constructs from Engineering to Biological Optimization Aspects. This PhD thesis was developed in a double doctoral degree program between Maastricht University and the University of Campinas in São Paulo, Brazil. The human body is a complex structure composed of different types of cells, tissues, organs, and systems. And when we have a failure or a malfunction in one of these systems, we need to develop strategies to restore and to repair these tissues. The tissue engineer triad, based on the use of biomaterials, cells, and biochemical tools, can offer these devices and constructs that will be able to repair the damaged tissue. Among all the tissues of the human body, the cartilage tissue is special and is of great interest because of the low self-repair capability, the limited vascularization, and because the lesions progress in an irreversible manner. The non-surgical treatments are currently limited, and the only cell-based approach approved in 2019 is the chondrocyte transplantation. The osteoarthritis is one of the most frequent cartilage disease reported, and as we can see, let me just get the pointer. As we can see here in a healthy knee, we see a smooth cartilage with no fusioning, a smooth bone. And as the disease progresses in this mild stage, the cartilage starts breaking down until we see a really severe stage when we have the bone moving against bone, and it causes a lot of pain. A potential alternative to build cartilage constructs is through the differentiation of stem cells, which has a high chondrogenical potential and is highly inducible in aggregate cultures. So as we can see here, we can culture the stem cells in monolayers, but we can also culture them in aggregates. And then when we have a cluster of cells typically formed by allowing a cell suspension in a droplet of media, and the final structure is spherical and uniform, we can call this cell spheroid. The spheroids can be applied in the field of medicine in different applications, starting as study models, especially for drug screening and pathological studies, but also as animal testing replacement, and more recently, as tissue building blocks to develop the tissue engineering in a large scale, the biofabrication. Also, we can use spheroids to promote automation and a feasible scalability, which is the topic of this thesis. We use this through bioprinting, which is a technique that enables automated deposition of cells and spheroids in a layer-by-layer deposition with height control and reproducibility. Here, as we can see, the spheroid-loaded bioink, we use the spheroid as a building blocks to construct a larger tissue. So also we can have the freedom to build complex structure in different geometries. Considering the fact of having this spheroid-lowered bioink, the spheroid I already presented, but also we can have a hydrogel component, which will be important to act as a support material, allowing cell proliferation and differentiation. Also, a hydrogel is the main component of the fluid phase of the cartilage tissue. In this work, two natural and biocompatible polymers were chosen. The first one, Alginate, which is extracted from brown algae and is the most frequently investigated classes of bioink. And it has an efficient cross-linking strategy. Along with Alginate, we chose to work with xanthan gum, which is a gum extracted from the bacteria Xanthomonas campestris, which is not yet frequently explored as a bioink, but it shows a height, viscosity, and stability. So considering all these concepts presented until now, the present thesis aims to answer the main hypothesis. Is it possible to use a bioprinting technique to manufacture stable constructs composed of stem cell spheroids suspended in a hydrogel, which could be subsequently differentiated to form a cartilaginous tissue? To answer this main hypothesis, the thesis was divided in 6 chapters. So the first one was a literature review in which we present all the aspects involving the spheroids and the main parameters involved in the high throughput production of these spheroids, along with applications and characterization. In chapter 2, we develop a spheroid device to be able to have a spheroid with a height control of diameter and geometry. In chapter 3, we first develop 9 formulations of a hydrogel containing the xanthan gum and the alginate. We mix this hydrogel with the spheroids and then we bioprint them. And in the sequence, we perform the chondrogenic differentiation to achieve a cartilaginous tissue. And in chapter 4, we study the migration, the fusion, the movements of these spheroids inside this hydrogel. And finally, in chapter 5, we made a cost analysis of the device developed here to produce batches of 100 to 5,000 spheroids to analyze the feasibility of using this in a cell culture lab, but also in a small startup company or commercial fields. And the chapter 6, in which we discuss and summarize all the topics presented until now. So as the main goal of the chapter 2, we developed the spheroid device to transform these low quality spheroids in spheroids with good quality, good sphericity, and uniformity in diameter. The technique was composed on 3 steps. The first one, we developed a 3D CAD model. We printed this using a 3D printer, and we finished with a device with 2 pieces that we connected to each other to have the full device. Then, using a non-adhesive hydrogel, we poured that hydrogel there to acquire that desired shape, and we had at the end a really flexible micro-molded hydrogel. In the sequence, we harvest the cells from a 2D flask, the common one. We seed the cells in the micro-molded hydrogel, and after some days, 5 or 7 days, we could see a lot of spheroids being forming there. And then we could also analyze this in a live cell imaging in a really efficient way. We validated this device using 3 types of cells, human dental pulp stem cells, mesenchymal stem cells, and also embryonic stem cells. Then in the chapter 3, we started with 9 formulations of xanthan gum and alginate. In that 4 formulations that did not flow after the inversion test, showing that they were good to bioprint, we did some characterizations regarding printability, regarding porosity, and regarding rheological aspects. Also, as you can see here, that's the fiber of the hydrogel and how the spheroids are organized there. And this is what a 4-layer printed construct, how it looks like. In the sequence, we wanted to analyze if we are printing spheroids with 5 days or 7 days. Then we analyzed the structure, and we could see that spheroids of 7 days, the structure was much more tight and compact, and we already could see the production of collagen. But then in the 5 days, we could see a higher metabolic activity. So we chose these ones to keep going with the study. Then we embedded these spheroids in the hydrogel, we print them, and then we analyzed during 28 days, and we could conclude that A high cell viability was observed in fully encapsulated 5-day spheroids on the printed constructs throughout the 28 days in culture. In the sequence, we analyzed the chondrogenic differentiation. So first, we start with collagen 2, which is the main marker of chondrogenesis, and we can see here that it was expressed. In the sequence, we analyzed the production of glycosamine glycans in day 1 and day 28, as you can see here in blue. So we could see a higher expression after the differentiation process. We also analyzed another more collagens, such as collagen 2, 4, and 10, and then we could see that collagen 1 and collagen 10, they were present in the whole process, in the beginning, day 1, and after the differentiation process. On the other hand, collagen 2 and collagen 4, they were not present in the beginning, but they were producing during the differentiation process. So we concluded that bioprinted constructs were efficiently differentiated to cartilage tissue, resulting in a hyaline-like pattern. And the constructs were viable and stable up to 56 days. And this is what I imagine a video of the cartilage constructs obtained in the presentesis. In red, we have collagen 2, and blue, the cells, and in green is the F-actin that we have more in the surface. In chapter 4, we analyzed how these spheroids were moving or behaving in the hydrogel. So we could see conformation changes to spheroid from ellipsoid in a migration or in the absence of a migration. Also, we could see the fusion of closely dispersed spheroids. In this study, considering 2, after 46 hours, they were completely fusion and forming only one structure. We also analyzed it with incorporating 5 spheroids, and here 13 spheroids, we could see a big microtissue at the end. Then we also could analyze 2 contrasting behaviors that was happening. They were really interesting. We can see here a spheroid in a hydrogel. We can see some clusters of cells detaching from the surface of the spheroid and going towards the hydrogel. It's interesting that on this case, the cell clusters stayed around on this portion, but in another moment, they come back to the main spheroid and they were incorporated there again. So we could see 2 different behaviors. And finally, in chapter 5, we analyzed our system 1 developed on this thesis, and more 3 systems already available in the market. We could analyze in a scenario of producing 100 of spheroids, 1,000 and 5,000. And then we could see that the device developed on this thesis show it to be feasible and cost competitive to produce all these 3 batches that we can use in a cell culture lab or implement in a small company or a startup. As a conclusion, I hope that I could see the history behind this thesis, and then the fulfillment of all the proposed aims was crucial to corroborate the main hypothesis of the present thesis. So it's possible to produce, it's possible to use an extrusion bioprinting technique to manufacture stable constructs composed of stem cell spheroids and a hydrogel, which could be subsequently differentiated to form a cartilaginous tissue. So with that, first I would like to thank you all for being here. I would like to acknowledge the funding involved in this project from the Netherlands and also from Brazil. I would like to thank all my supervisors, Professor Lorenzo, Dr. Carlos, Professor Angela, and Dr. George. I also would like to thank all the collaborators that made all this possible. And also now, I would just like to say 2 phrases in Portuguese, if it's okay. Gostaria de agradecer à Faculdade de Engenharia Química da Unicamp e a todo mundo do Brasil que está participando e assistindo, especialmente a universidade pelo apoio. And my family, my mom present here, thank you. And my family watching in Brazil. Thank you very much.
Thank you very much for this clear presentation. We will now start with the opposition, and the opposition will be opened by Professor James Yu. He is Professor of Tissue Engineering Technologies at Wake Forest School of Medicine, Winston-Salem in the United States. Professor Yu, the floor is yours.
Well, thank you. First, I would like to congratulate for doing such an enormous work, excellent work, and there really wasn't too much that I could criticize or comment on. The thesis is very well thought out, and very well written and well read. So I really enjoyed reading your thesis. Again, congratulations. I only have a few questions, and those are not as specific, but in your introduction, presentation and in your thesis, you describe about the tissue challenges and talk about osteoarthritis and the difficulties and challenges that we face in general to repair and treat those patients. But the rationale for developing a bioprinted chondrocyte-based spheroid for very specific application was not really clear, and I was wondering if you're aiming and targeting to treat osteoarthritis or any other disease conditions, or if you had any specific conditions that you had in mind. And one question that I have is, if you target specific pathological conditions, then how would this spheroid platform or system would be used for translation?
Okay, thank you, highly esteemed opponent, for your question and your kind words. I believe that we chose to use the spheroids because we can have there much more cells than we use in a single cell scaffold, for example, and especially because the cells are cultured in a close contact, so the tissue is a bit more mature if we start thinking of only working with single cells. In the beginning, our idea was to use this to shorten a bit the time to have a construct to try in a patient, for example, because our idea was to print the spheroids on the gel, to perform this chondrogenic differentiation for 28 days. But our idea is to see implanting in animals first, of course, if the time to finish the process will be done in the patient. So we are trying to develop a more shortening technology. So we are targeting, yes, to try in a patient, but we chose osteoarthritis, especially because we are differentiating to cartilage. And is that one that is needed most.
Right, but may I ask you a follow-up question on that? So, as you mature the spheroids in vitro, these cells do adhere and produce extracellular matrix that minimizes or decreases the chance of diffusion. So evidently the cells in the central core tend to necrose. You can develop a good spheroid outside the body, but you cannot avoid the inherent problems or challenges that we all face when we transplanted the spheroid back into the the body. So those have to be well thought out. So the next question is, in your chapter 5, you discussed about, cost analysis for producing these cell aggregates, and you have compared to, other available methods. And, that, chapter was kind of out of focus. And so what are your thoughts? What was your motivation for, touching on the cost analysis?
Yes, the motivation was because when we developed this device, we were looking to scale up the production. So we started production in the lab, for example, 100, 200 spheroids to print small constructs. But at the end, I was producing 4,000, 5,000 spheroids to print several constructs to perform all the characterization. So then we started thinking if the device that we were using was good for that. And then we started thinking maybe we could try to make this in a different way, because it was a bit of hard work in the lab to produce that amount. And then that was the question that triggered us to try to see what we have in the literature, to also try to improve our method. And then we ended up finishing with the business case, the cost analysis study.
No, that's very good. Good answer. However, these are, patient-specific cells, right? So, obviously you cannot, unless you're thinking of using allogenic or xenogenic, purposes, but then, these, I would assume that, any application in the body, of course you can mass produce, for, testing drugs or developing drugs or, toxicity testing. But if you were to implant it in the body, it will be patient specific, applications. So you would only need, certain amount of cells or, spheroids, right?
Yes, I agree. We also discussed a bit on this chapter that the application is really important, because if you are, for example, applying to study pathologies or drug screening, we don't need that amount of spheroids. But if we are going to bioprinting, if we are indeed using them as small tissues, like building blocks, we will need a huge amount. So also the quantity, and also if it's specific for a patient. Also in chapter 2, we try to use another types of cells because of this, to see if the cells will behave in a similar way or not, to try to arrange the most possible the possibilities.
Okay. My last question, I think, the process or platform technology that you have developed, is a, promising strategy for cartilage tissue engineering. It would have been, this thesis would have been stronger, if these spheroid constructs were put to work, and to demonstrate the ability to build tissue construct or validate in applications. Of course, this doesn't mean that you should stay and, do all this, because you gotta get out and graduate. But if you were to have more time to do so, what study will you perform to demonstrate the utility of the bioprinted spheroid?
Yes, really interesting question. We were thinking a bit on that. Something that we would add in the study will be co-culture some vascular progenitors together with our HMSCs, because we know that the cartilage is avascular when it's adult. But when we have an immature cartilage, is what we do in the lab. We have some vascular progenitors to literally opening the channels there to promote the diffusion of oxygen and nutrients. So we were thinking on that, to try to combine some vascular cells, to try to print as much as possible these different constructs, and to see indeed which one will be more promising in the future, thinking in clinical trials and application.
Very good. Thank you so much. Wonderful work. Thank you.
Thank you. Thank you very much.
Thank you, Professor Yu. The opposition will be continued by Professor Marisa Masumo Beppu, Professor of Chemical Engineering of the University of Campinas in Brazil. Professor Beppu.
Thank you very much. I would like to begin thanking both universities for inviting me for this board. And it's also a pleasure to see how the candidate has developed even the text and also the presentation. I think both were very clear. They have shown big amount of information, of insights that are valuable for this field. So I think there are lots of interesting material as building blocks for the state of the art in this field. So I would like to congratulate all the advisors and everyone that was involved in this very interesting piece of study. Monize, I just have two questions, but probably you are going to take a little longer to answer some of these because they are much more, let's say, fundamental. Because I think although the objective is to have an artificial organ or something that we want to build as a construct from these spheroids, I think the insights that you brought to knowledge about these systems, how they evolve, how they have the motility, how they fuse, are the most interesting information. So if you think about what you have learned about these systems and you think about application, which would be the main challenge that you still have to go through to see something being directed to a patient? This is the first question.
Yes, highly esteemed opponent. Thank you for your kind words and for your question. I believe that, wait, let me organize my mind. Sorry, can you repeat the beginning of the question again?
Yes, sure. The main challenge that you would list to go through to have a product or something to be implanted in a patient.
The main challenge I see when we were choosing which hydrogel we are working, and also the spheroids, a challenge that we saw was how viscous, for example, the hydrogel should be, because it was something that we were discussing a lot, that we want something printable and that maintain the structure after printing, but we can also we cannot have something really stiff, otherwise, as we show in chapter 4, the spheroids will have some difficulty to walk and indeed to move and fusion to each other. So I think to find exactly the spot, like the balance between the viscosity of the hydrogel and the amount of spheroids and how they can move, I think was one of the biggest challenges that we face it here. How to implement the bioprinting in a satisfactory way, but also how to make this possible that the tissue will indeed fuse to form a bigger tissue. Because if we don't see the fusion, we don't have a micro tissue at the end. So I think this was of the main challenging, and also how to permit the diffusion in a better way. Because if we are thinking have a big construct, as I told to Professor, we could put some vascular cells to start building some small channels, and then I think we are going to construct a bigger construct. But these two challenges, I think, were something that we needed to work a bit more and try to develop a better way to indeed scale up this production. Otherwise, I think we will be printing small things and small implants. I think these both factors I see as the challenges. I don't know if I answer.
Yes. And not only answered, but also gave me the link for my second question. If you think about the two components that you have cited, which are the matrix and the cells. And I think that one thing that you wanted as a good output is the fusion that you have seen. This was something good for you to have conditioned to build the organ or the construct that you wanted. If you think about this output as a fusion and forming a bigger construct, how would you rank as in terms of importance for this phenomenon, you would rank first the matrix as something important, or the cells are the major, let's say, responsible for this? I know that it's a tricky question, but I wanted to see your discussion towards this, and even with different cells that you have mentioned in your work. So I wanted to hear a little bit about that from you.
Yes. I would say that the cells are more important because they are responsible to produce the extracellular matrix. So if we try to organize better the timing to the hydrogel to degrade and to the spheroids produce the ECM, if we can combine these curves in a better way, I think is the ideal, because we are working with biocompatible and biodegradable hydrogel. So after some time, we don't know exactly, but after some months, the hydrogel will not be there anymore, but the spheroid should be strong enough to produce the ECM to replace that gel. So I think the combination of both, but I would say more the cells, because they produce the ECM.
And probably that's why it can be very particular for each kind of application, right? Okay.
So each type of cells that we are using.
Okay, thank you. I'm very glad with your answer. So that's it. Thank you very much.
Thank you.
Thank you very much, Professor Beto. The opposition will be continued by Professor João Mano, Professor of Biomaterials and Chemistry from the University of Aveiro in Portugal. Professor Mano, the floor is yours.
Thank you very much. So first I would like to thank the institution and the supervisors for inviting me for this ceremony and for this very interesting committee and this very interesting thesis. So also I'd like, of course, to compliment the candidate and all the family and their friends. So I believe it's a very, very important and very nice day for Monize. So I would like to highlight what has been mentioned about your thesis, Monize. So it's really interesting work with a high level of innovation, very multidisciplinary, really in the front area of different areas related with biofabrication, bioengineering, and regenerative medicine. So congratulations for that. So I have a few questions much more on the technical side. So the first question is related with your decision in terms of how to incorporate cells with the matrix in terms of the differentiation. So in your cases, so what is really the advantage of integrate the stem cell spheroids and then differentiate inside the bioprinting construct instead of pre-differentiate the spheroids into microcartilage pellets and then integrate them into bioprinting constructs? Because in the second possibility, I believe, but maybe I don't see the full picture, you will have much more control on the differentiation medium, diffusion, for example, of the differentiation factors, dynamic condition, that can be a little bit more difficult when you want to differentiate the stem cells already in the constructs.
Yes, dear highly esteemed opponent, thank you for your question and your kind words. It's a really interesting question. I totally agree. When we are thinking, when we started the differentiation, that was our main question. Should we differentiate the spheroids before and then mixing the hydrogel and print, or we should use stem cell spheroids, mix in the gel, bioprint, and differentiate? But since we were analyzing the process in the whole chain, we would like to have a more genetic and a more versatile construct. So we prefer to work with stem cells because our idea was try first chondrogenic, but for example, this opens a lot of possibilities to try different differentiation medias. So we can try this to osteogenic, we can try to vasculature, we can try different conditions. And if we use the chondrogenic media before, we are finishing the process with a really specific tissue because we will only cartilage, which I tried here. But our idea was to open this a bit more, like to indeed offer a process, a genetic one, to try what we want. And also I agree what you told about, because it's different, of course. It's more difficult to perform the differentiation inside the hydrogel because the diffusion is a bit compromised. We don't have the same distribution of oxygen throughout the whole construct as we have using media. So we have this barrier to face, but at the end we could differentiate. We were also thinking to increase a little bit the concentration of some specific components, for example, TGF-beta or dexamethasone, to save that delay that we are having with the diffusion in the hydrogel, let's say, but at the end it was not necessary. So we went to that direction to have a more versatile bioink.
Thank you very much. So I would like to invite some of your para-nymphs to read proposition 6.
A hydrogel formulation composed of xanthine-gum and alginate proved to be capable to support efficient spheroid encapsulation, differentiation, and stability in culture. 6. Yeah. Sorry, life is 3D.
Okay, thank you very much. Okay, so you want to create 3D tissues or life combining cells and hydrogels. Usually people do that by dispersing free cells inside the matrix. Okay, but in your case, you decided to incorporate cells previously aggregated in spheroids, okay, which is not the case when you look, for example, cartilage tissue, when you see chondrocytes very well dispersed. So it seems a little bit contradictory why you are joining first the cells in order to have something where the cells are dispersed natively.
In the beginning, when we were studying the structure of the cartilage tissue, also we observed this because they are dispersed and also they are completely different in the 4 phases of the cartilage. So we have more chondrocytes in one phase, less in the other. They are organized in different orientations along with the collagen. So, but we choose to use the spheroids to have a bigger amount of cells in a more mature way, even though we see that we don't have a lot of cells in the cartilage. But also an interesting thing is that when we see this image, normally we see a mature cartilage. So the cartilage was already developed after years and years of maturation. But probably in the beginning, a really immature cartilage, we had much more cells. Otherwise, we could not have a lot of ECM or a lot of proteins produced by them. So we were thinking to try to reproduce a more immature cartilage, giving more quantity of cell as possible, even though we see that the adult one doesn't have the same pattern.
Okay, and the quantity of aggregates that you should put in your hydrogels, from the image, we see a lot of very dispersed aggregates. So what do you think will be the limit, the minimum amount of spheroids in terms of density to generate a completely formed cartilage in all the volume of your constructs?
That's a really challenging question, Professor, because we were thinking to print as much as possible. But also we have some problems with the printing because a cell, for example, has 20 micrometers, but a spheroid I was using 200. So we have some problems also to pass to the nozzle of the syringe and to print. And also they could fusion or not. So, and we couldn't control a lot the distribution on the tissue. Ideally, we would print as much as possible to offer a really big pool of cells to enable them to fusion. But I think the main idea was to try to find this number, how many cells we need. And also something that we are trying to find in chapter 4, which distance we should put both spheroids that they will be able to fusion. Mm-hmm. So 200 micrometers, 300 micrometers will be enough. And then we could plan this in the cut before to try to reproduce that. So that will be the main question, but it's a bit hard to define this number.
Okay, thank you very much. Again, congratulations for the beautiful work. Thank you.
Thank you, Professor Mano. The opposition will now be continued by Professor Tim Welting. He was the chair of the assessment committee. Professor Welting is professor of molecular cartilage biology at this university. Professor Welting.
Thank you. So first, of course, I would like to congratulate you with probably something which is your life work. So and also your team, very well done. And it's always a pleasure to dive into a topic that is just a bit off my topic. So that's really interesting. So and of course, you start with your thesis by looking at the cover. Mm-hmm. At least that is how I approach a thesis. And then I see calcium ions. And actually, this is where my first question will start. Because in my world, well, calcium ions and cartilage or chondrocytes is a very bad marriage. As in, well, hypertrophic differentiation, mineralization, calcification, these are things that you really do not like to have in your cartilage. But of course, the alginate that you use, well, you use quite an amount of calcium actually to polymerize your alginate, right? So it's 125 millimolars, if I am correct, for 10 minutes. So do you think that is still a good marriage between the calcium, or maybe it is just the amount of time that these two are married? Is that? So could you please start with that?
Yes, dear highly esteemed opponent, thank you for your question, your kind words. Actually, we could have worked with strontium, for example, or other bivalent ion. But normally we work with calcium because it's the most known cross-linking strategy when we are working with alginate. So we went to that to be in a more safe place, let's say, because the Xantangan was already a big novelty for us that we didn't know exactly how to try to cross-link because we don't have, it's not really well known as we have for alginate. Yeah, so, and then we could try other ions, sorry.
So you're actually saying that because it has been done before, this is the way how you do it. So
no, no, I totally agree, especially because if we can have some ossification, the calcium will not be good. But I think that we perform the cross-linking for 10 minutes only, and then we wash that solution. Was really diluted the solution. It was just performed once in the real beginning of the process. So after that, we differentiate for 28 days. So I'm washing with the media culture even every 3 days. Yeah. So we believe that this calcium will be washed out or will be in the middle of the molecule of the alginate.
Right. Yeah. So and then if I then tell you that I just checked with my PhD student that, for example, if we use 6.6 millimolar of calcium chloride, just with chondrocytes, they start to mineralize within 2 days. And then, so 10 minutes seems really like a short time, but also cellular signaling events can start. So it's I do understand that it is like almost unavoidable. But so did you ever check what this amount of calcium does to the chondrogenic differentiation potential? So have you ever checked, like exposing them? Yes or no, and then look what it does?
We try to expose in the beginning, before starting with calcium, I try to expose the calcium chloride with cells in different conditions and different concentrations, but I was just able to check cell viability. In cell viability was okay, the cells are completely the same, but I was not working with animals or something more complex, only cells.
Okay, because that leads me to the rest of my series of questions, which is more related to parameters that you tested. Yes. And one of the things that really struck me, right, is that you already have collagen type 10 already present at day 1 in your spheroids.
Yes.
And I'm just wondering, could that be a result of your calcium exposure? Because MSCs really normally do not express collagen type 10 to the extent that you're showing.
Yes, yes. We could be, I think, because we could see in the beginning, day 1, and also we could see that was maintaining, that concentration was not increasing, but was already there since in the beginning.
Okay, so I think that's enough said for the calcium. So going to some other parameters. So you're using things like maturation of the construct. Could you define what you actually see as maturation? So what is maturation of a cartilage construct? What does it involve?
Yeah, the maturation for us is to see if the tissue was producing a lot of extracellular matrix, for example, if the cells could be alive for longer periods. Because of this, we made a pilot study with 56 days to see if we could make the cells viable until there and producing a lot of ECM. Mm-hmm. We consider this a maturation in a lab environment, of course, but it will be interesting to test this in an animal or in a clinical study.
But maturation can also be like collagen content versus gags or cross-linking or these kind of things. Is that something that you would consider?
We consider maturation as the way that the tissue can remodel and can be forming throughout the time analyzed. So yes, I consider also the production of GAG and collagens. Yes.
Okay. And then one of the readout parameters that you use quite often, and I think you already mentioned them, is viability and metabolic activity. And if I got it correctly, you're measuring metabolic activity by actually measuring ATP via a luciferase-like reaction, right? Mm-hmm. But what does metabolic activity actually mean in the context of a chondrocyte? So is a chondrocyte a low or a high metabolically active cell? And so I'm really looking at, because you see big differences. Yeah. But what does it mean in the context of your...
Yeah, in the context of my thesis, we were analyzing the ATP expression because we were exposing the cells to severe conditions. For example, the shear stress that we have when we print, also the cross-linking, also the exposure, the cells for longer period. So these kind of conditions, we would like to see if the cells will be able to be producing ATP in the same rate from after all this stress.
But I'm looking actually more on, to a answer in the direction of is high good or low good, for example. So what are you looking for? Are you looking for a high metabolically active cell? Or
yes, to be honest, I was looking to the spheroids to maintain that viability, not to increase, but to be able to suffer that process and to be able to stay there and keep going alive. So we are looking to maintain. But of course, if we can increase, it will be better because it can also mean that we have a higher ATP, so the cells are comfortable to keep growing and to keep going to the maturation.
Did you ever try to achieve some reference point? For example, if you take chondrocytes from a human healthy individual, for example, measure ATP levels, that you have an idea of what you would be aiming at?
I haven't done that, but this is a really interesting thing that we could perform, especially with chondrocytes, to see if we are reaching the same. Yes, it's an excellent idea, but I haven't done that.
Yeah. Do I have time for one small question?
Yeah.
Okay. Because I was also interested by, so also you already mentioned this during your layman's presentation. You pre-made those spheroids 5 or 7 days. Yes. And then you did all the measurements and you chose, if I'm not mistaken, the 5-day condition. Yes. So did you also check other time points? A simple yes or no would suffice?
Yes, I tried with 3 days, but they were not ready yet. They was assembling in single cells, so I couldn't use as a full structure.
Because I was interested in knowing whether, this pre-maturation period would influence, for example, the chondrogenic differentiation capacity within your construct.
Yeah, I think if it's too intense, if the aggregation is too intense, maybe yes, it can affect. But I think in the way that we were doing just to aggregate the cells, I think no. I think depends of the time. If I keep going that system for like 5, 7, 10, 12 days, I think it's a problem. Mm. But for 3, 5 days, I don't think it's a problem.
Right. Okay. I'm really happy with the answers to my questions, and I give it back.
Thank you, Professor Belting. The last opponent of this session will be Professor Martijn van Griensven. He's Professor of Regenerative Medicine at Maastricht University. Professor van Griensven.
Thank you, Mrs. Prorector. Dear candidate, muito parabéns for your excellent study, for your very nice booklet, also to the supervisors here in Maastricht and also in Brazil. I really enjoyed it, and I wanted to actually ask a lot about the cells, but it has been done already. So I'm going more into the chapter 4. You see that there is an ellipsoid formation. So if you write nice round spheroid, getting more ellipsoid. And you discussed that it may be because of oxygen or nutrition gradients. I was thinking, when I saw that, I was thinking, well, can it be also some mechanical components? So like a load, a burden.
Yeah, dear highly esteemed opponent, thank you for your question. I think yes, and also I think these mechanical compounds can be also helping the spheroid to move. We made also an analogy with amoeboids or something like that, the way that they move, because they don't have arms and legs, so it's a full structure. So maybe if we have mechanical compounds in there, I agree with you that maybe it's because of that. We also thought that oxygen and nutrients is the main cause, because we have a big matrix, but we have a lot of cells there also. So probably the depletion of nutrients was happening around, and they need to.
And did you see that? Did you see then that it was all going in the same direction, or was that?
No,
chaotic?
was a bit random, and also that to contrasting behavior that I showed, we were really surprised with that, because some cells were going to completely random directions and coming back or not, so we couldn't see. But at one study, to be honest, we see that when we have, for example, 2 spheroids, we could see by eye that they are kind of going to each other, but I cannot affirm this without performing more experiments. So was random, or maybe some cell signaling could be happening there also, which is difficult for us to assess in the way that we are performing now, but will be something to look into.
Yeah, because that's very nice what you say. So the movement of your spheroid or is of course an interaction, like Professor Beppel was also telling, between your extracellular matrix and your cells, right? So there's adhesion molecules and so on. So do you have an idea which adhesion molecules or which partners ligands in your ECM play a role?
Maybe the molecules of the cytoskeleton. I don't know if I pronounced this correct. Sorry if not. But I think, yes, these molecules maybe will be interacting with that ECM, interacting with the cells. They were communicating to be able to make a plan and move to some direction. I think, yes.
And do you think you can then even change that? So if you want, for instance, like, because you have been speaking also about other cell types, other things, when you want more movement, for instance, or a different movement, do you think you can then actually modify that with your, either with your ECM, with your materials you use, or do you have any idea on that?
I think yes, but I think it will be interesting to analyze the same behavior with different cells, for example, that we have different ECM being produced and in different concentration, also in different hydrogels, because I could just see this in one system, one type of gel, one cell. So we are a bit blind. We could be able to see more. So maybe yes, I think we've tried with other cells, we will trigger some other mechanisms.
And could you also add some ligands that make it more sticky or less sticky?
Yes, I think also yes. And also, I don't know, something to make that more flexible, or making some channels in the gel was something that we are also thinking, to try to leave the space open to the spheroids to be able to move, or something extra. Mm-hmm. Everything to try to able the movement of them. Yes.
Okay. You actually described like 4 modes of different movements, right? So you had these big things, you have cells going out and so on.
Yeah.
What do you think was the most important movement in your system?
I will say 2 movements, but I'll explain why. The fusion, I think, is the most important, because is indeed what we want. We want the spheroids to fusion to make a bigger tissue. But this we also have seen in the literature in many studies. So I think that the cells detaching from the surface and migrating in the spheroid is the most novelty that we are bringing, because we see different behaviors, and then we couldn't see another study in the literature reporting the same thing using healthy cells. However, we saw with cancer cells, for example. So it's something that we are trying to study, to correlate, to see if we can find a mechanism behind that we can compare.
So, and when you say the cells moving out, is that a migration, or is there another mechanism what could play a role in that sense?
I would say migration, but not the full structure. It's a cell clusters that detach and really migrate around. But I would consider a migration,
And
like it populating other sites if possible.
wouldn't be at a certain moment then your spheroids that you added in with your bioprinting be depleted? So when the cells go out, then at a certain moment, well, what is left over?
Are you saying like I have a pool of cells and they need to?
Yeah, so you have your spheroid.
Yes.
And the cells move out. You just said you think they migrate out, right?
Yes.
So your spheroid gets smaller.
Yes.
Yeah.
Okay. Yeah, I see.
So at a certain moment, so your cells have moved out,
and I don't have
you
this
don't have the spheroid anymore. Do you think that happens, or are there other mechanisms that may be preventing that?
I think that would be more that they are growing and being able to release some pieces, let's say, to be growing, but I don't believe that they will completely disappear. I think because it's a main structure at the end, so I think they will be able to release some.
Do you think there's proliferation involved?
I think yes, but not in the core of the spheroid. I think this is more on the surface. Because of this, I also believe that once that they are detaching, probably they are the ring of proliferation on the border of the spheroid, not that one in the core.
Then I may come back to what my previous opponent said.
Yeah.
So he was telling actually, okay, are they now differentiated? Are they mineralized? Would it influence that?
I think yes, because also we need to consider that we have a gradient there about everything of nutrients, of oxygen. So I also believe that some of them can be mineralized, some of them are going towards chondrogenesis. So I think we have a diverse population there.
And when you want to have a mature tissue, so like the first opponent actually asked James Yu, he was asking you, okay, for what disease is that? Would it be beneficial to have like a population that is not very homogeneous?
I think considering in the last step of the chain, like to build, for example, an organ or system that we have indeed different types of cells, different populations, I think is good. But in the beginning, now that we are going to develop a process to try to understand, it will be better if we have more homogeneity. But in the real scenario, let's say, I think we will have subpopulations. We cannot control really precise because of these gradients. So I think it's a bit difficult.
And do you think your cells would even leave your hydrogel? So when you implant it, for instance, or is this movement, what you have been describing, really inside the hydrogel, going inside and not going outside, or what do you expect?
To the body, you're saying the surrounding. Yeah. I think if we needed to analyze if the both tissues, they have the stiffness and the viscosity enough, because I think if the hydrogel is not stiff, but the tissue is stiff, I think the cells will have some difficulty to penetrate and to keep going. So I think that if we can also make this more similar, like in stiffness, especially in viscosity, I think yes, I think they will be able.
I think you also use different platforms like adhesive cell culture plates, non-adhesive cell culture plates, right? Did you see on the adhesive cell culture plate more cells that came out of your gel actually, or?
No. Normally they were really embedded, really immersed in the hydrogel. No.
So they really don't go out in that sense?
Yeah, in that sense, no. They were more keeping the,
And when you start playing around with your spheroids in different amounts, do you think when you fill up your bioink in that sense, would still these movements take place? Or is that a certain moment like a kind of contact inhibition, or are they influencing each other?
Yeah, this is an interesting question because we observed this phenomenon in the beginning of the culture, so in the first days. I don't know at the end how it will happen. If the bioink will start degrading, maybe this could be something.
You want to finish your answer?
I think I finished it. Yes.
You finished. Okay. Monize Caiado Decarli. The time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await results of our deliberations and our return in this room.
Please, all rise. You did it. Oh my God. The presentation was good, right? And they like it a lot. Thank you. Let's give my hug to my
Go.
Monize Caiado Decarli. The degree committee here present, here and online, has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Moroni is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent and independent, and responsible?
I do.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Monize Caiado Decarli, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
I now give the floor to the supervisors for the laudatio.
Dear Monize, dear Dr. Decarli, what a journey. It seems that we started this journey a decade ago. Between your first email requesting to do part of your PhD here with us until the date you finally arrived, it seemed to have taken ages. After securing your funding, you have finally arrived in September 2019 for an 8-month visiting PhD student. The 8 months quickly became 15 as COVID started to play a trick on all of us. With the impossibility to travel, you continued your journey, but instead of going back to Brazil, the farthest you could go was to the Merlin labs. It was an incredible period where you navigated through adversity seamlessly without a single resting time to make more and better. The product of that effort was shown today, but it will continue for months and years to come. Monize, what an incredible scientist you have become. I still remember the first months where we spent endless hours together in the lab, but after, during the discussion of the results. Frequently, you would share your results and you would ask me if I was happy with what you have achieved. I would always quickly return the question to you, as it is important that you are first and foremost satisfied with what you have accomplished. You would then discuss the small details, which in some cases were small only at first. The most puzzling of those moments were when you all of a sudden attempted to change your material. Carlos, vamos mudar essa chantana? Carlos, we need to change this chantangam. With some patience and reasoning, we decided to stick with the plan and keep working on the material that you have been working for months. After some fine-tuning, you were able to obtain bioprinted constructs, and then you went on with the differentiation studies. COVID was being restless with humanity, but so were you with your cells in the lab. You were the commander-in-chief, but cells have always something to say. As it would be expected, Monize was always a great lab citizen. As soon she started to build her own network of friends in the institute. Everyone was impressed how a PhD would always have a happy smile, and this is one of the things that everybody would always recognize on you. No matter how bad the day was, no matter how harsh the cold winter was on you, Monize would always be there smiling. Some words from your supervisors in Brazil. An important part of this thesis where you acknowledge everyone who gave you support to carry out your thesis, whether technical, scientific, and emotion. In this regard, the 4 pages of acknowledgements clearly show the power of setup network, captivate, delight, and benefit from the best people you have to offer. And more than that, to realize, to really recognize the added value of contributions of each one to your work. Your dazzle minds and hearts always achieve noble goals. Your power to fascinate students and colleagues, also formally offering your knowledge with great generosity, is also a great asset. Now in Portuguese. Monize, nós nos lembramos muito bem de quando você fez os primeiros contactos iniciais connosco, já com uma clara visão de desenvolver um trabalho de alto nível em colaboração com grupos estrangeiros de grande contribuição na área de pesquisa que você vislumbrava abordar. A parceria de orientação no Brasil não poderia ter sido mais aprazível e profícua, e a sua tese muito contribuiu para estreitar os laços entre a Unicamp e o CTI. Foi também uma enorme satisfação dividir com os nossos carinhosos parceiros da Universidade, caríssimos parceiros da Universidade de Maastricht, a tarefa de guiá-la em seus estudos. Muitas horas agradáveis de discussão prolongada e proveitosa, muita troca de complementariedade de ideias na análise dos seus artigos. Você realmente desconhece os limites e é capaz de se superar a cada momento. É uma honra trabalhar com uma pessoa como você, agradável, proativa, comprometida e perspicaz. During your stay in the Netherlands, it was clear that you matured even further in every way, from the technical to the emotional. Your resilience and proactivity resulted in excellent results shown at this moment in an unquestionable proof of it, and this moment is an unquestionable proof of this. Your work was evaluated by major world references on the subject of biomanufacturing. We are sure that when we suggested such names, you felt some apprehension, but at no time you showed fear or doubts because you trusted in the quality of your work and your supervisors. In these almost 6 years that we have the honor to meet with you, at no time we have seen you discouraged or sad. On the contrary, you were always tireless and had many ideas and many proposals for work. Having 4 demanding advisors and satisfying them simultaneously was not an easy task, but you managed in a brilliant, always with courtesy, competency, and work capacity, integrating ideas in the most qualified way possible. Achieving your doctorate is a logical sequence of the history of training and essential skill for an interdisciplinary area of biofabrication. So it will be your postdoctoral fellow position now at Merlin, but with even greater responsibilities. We hope you can continue to be an excellent interlocutor. Before we finish, I would like to thank the family. Quero parabenizar, now in Portuguese, sorry. Quero parabenizar toda a sua família pela formação, carinho, proteção e suporte que lhe permitiram ser o que você é a cada momento da sua vida. A presença da sua mãe nesta cerimónia será inesquecível para todos nós, mas em especial para ela e para você. A cada um que assistiu à defesa, agradecemos pela atenção e esperamos que, como nós, tenham desfrutado deste momento tão importante. Monize, que a vida continue recheada de muitas conquistas e felicidades. I hope I will finish soon. Let me take this final moment to thank all the members of the committee for taking the time to read critically your thesis and for being with us today. In particular, Professor Romano, Professor James Yu from the University of Aveiro and Wake Forest School of Medicine, as well Professor Marisa Beppu, University of Campinas, and Professor Martin van Greensen from Merlin. Sorry, it's very difficult to pronounce your surname. I would like to thank Professor Tim Wellington from our MUMC for sharing the assessment committee, the reading committee, and for the shared presence. Monize, I'm out of words, thank God. Rest assured that we see each other soon in the lab, even if it's just for a chat. It was a pleasure to meet and to work with you. More smiley days to come ahead. Thank you very much.
Thank you. Dear Dr. Decarli, also on behalf of the Board of Deans of this university, I congratulate you with the honor you have acquired, and I wish you a lot of success in your future career, and I hope you have good memories of this university and of Maastricht. And I also hope that you have a lovely day after this ceremony, because it must have been a lot of stress for you. but it's over and you obtained a degree. Congratulations. Also your family and friends and colleagues. I now formally close this session, which means that I ask